fast-acting vitamin D receptor agonists for the treatment of IACC conditions, infection-associated chronic conditions, calcitriol versus paracalcitol, Part 5. The links to the first three portions of this YouTube series are highlighted at the bottom of the slide. In Part 4 of this series, we discussed this news report from the Center for Infectious Disease Research and Policy, CIDRAP, at the University of Minnesota. This is from March 20th, 2026. Up to 60% of healthcare workers may have long COVID 4 years after infection. We demonstrate a nonlinear decline in PASC-specific symptoms over time, with up to 60% of individuals still reporting symptoms after a median of almost 4 years after ancestral SARS-CoV-2 infection. In Part 4 of this series, we discussed the impact of COVID on renal functions, particularly the ability of proximal tubules to synthesize and secrete the powerful immune hormone calcitriol, 1,25 vitamin D3. We also discussed the potential repurposing of a synthetic analog of calcitriol, paracalcitol, 1,25 vitamin D2, to replace deficient levels of calcitriol in long COVID patients. In Part 4 of this series, we also discussed this news report from DG Alerts in February of 2026. Long COVID tied to elevated depression, anxiety risk 3 years beyond infection. Adults experiencing long COVID may carry significantly increased risk of depressive and anxiety symptoms up to 3 years after initial severe acute respiratory syndrome coronavirus 2, SARS-CoV-2 infection, according to a population-based cohort study published in BMC Public Health. Reading from this news report, psychological or psychiatric conditions. Notably, long COVID was linked to a 2.6 higher risk of depressive symptoms at the 3-year follow-up, relative to no depressive symptoms at either visit, and a 2.5 higher risk of anxiety at both follow-ups relative to no anxiety at either time point. They're discussing people with long COVID versus controls. In Part 4 of this YouTube series, we discussed the newly discovered pathway by which activated vitamin D receptors can upregulate a key enzyme in the cytochrome P450 family. This enzyme is an epoxygenase. It's known as CYP2J2. It puts epoxy groups on arachidonic acid, converting this pro-inflammatory lipid into anti-inflammatory lipids known as EETs. And the upregulation of EETs in response to vitamin D receptor agonists may be a key therapeutic strategy to help resolve ongoing inflammation in the nervous system as well as the kidneys. And that connects to this key paper published in December of 2024. Persistence of spike protein at the skull meninges brain axis may contribute to the neurological sequelae of COVID-19. On the right, lower right, you see a mouse which has been chemically cleared so that the fluorescence from the internal organs can be visualized. When COVID-2 binds to the cell surface, there is an enzyme which cleaves the spike protein, releasing the spike protein S1 fragment. That fragment is highly toxic and can circulate throughout the body. Here, what they're looking at is a model of long COVID, and you can see the localization of the S1 fragment. It's present in the brain, the heart, and highly localized in the kidneys. Everywhere the spike protein is localized, these are potential sites for the activation of the innate immune system. Cells like macrophages react with the S1 fragment of the spike protein and enter a hyperinflammatory state. Persistent inflammation involving macrophages plays a central role in long COVID pathophysiology. So in discussing these phenotypes, the pro-inflammatory M1 macrophage, which infiltrates tissues and causes immediate and long-term damage, versus the anti-inflammatory M2 macrophage phenotype, which helps tissues heal. And one way to prompt the development of the anti-inflammatory M2 macrophage phenotype is to expose macrophages to EETs, epoxy-eicosapentaenoic acids. So the hypothesis here is that spike protein S1 fragment is likely to provoke chronic renal inflammation because of the high localization of the S1 fragment in the kidneys. And by damaging kidneys, particularly proximal tubules, where calcitriol is synthesized and secreted from, this animal is likely to acquire vitamin D resistance. And that's a common theme that we have been emphasizing: acquired vitamin D resistance from microbial infections. Now I would like to review a historical paper which indicates the utility of paracalcitol 125 vitamin D2 as a treatment for renal inflammation. And this paper was published in 2008, so nearly 20 years ago. Paracalcitol inhibits renal inflammation by promoting vitamin D receptor-mediated sequestration of nuclear factor kappa B signaling. And this paragraph is from the conclusion of the paper. In summary, we have shown in this study that paracalcitol attenuates renal infiltration of inflammatory cells and inhibits tubular RANTES expression in obstructive nephropathy. We will discuss RANTES shortly. This anti-inflammatory effect of paracalcitol seems to be mediated by its ability to induce the vitamin D receptor-mediated sequestration of nuclear factor kappa B signaling. Therefore, in addition to preserving tubular epithelial integrity by blocking epithelial to mesenchymal transition, as previously reported, active vitamin D, i.e. paracalcitol, also exerts its beneficial activities in chronic kidney disease by inhibiting renal inflammation through directly disrupting nuclear factor kappa B signaling. RANTES is also known as a CC motif chemokine ligand. What is a chemokine? A chemokine is an agent that promotes leukocyte infiltration and inflammation into tissues. So RANTES/CCL5 is a pro-inflammatory chemokine that acts as a key mediator in kidney damage, promoting leukocyte infiltration and inflammation in acute kidney injury, AKI, also in transplantation rejection, as well as progressive nephropathies, kidney disorders. It is upregulated in renal tubular cells, causing fibrosis and damage, making it a critical therapeutic target in autoimmune renal injury. So condensing this information into an information vector, paracalcitol 125D2 interacting with the vitamin D receptor inhibits nuclear factor kappa B pro-inflammation signaling. Normally, nuclear factor kappa B upregulates this chemokine, a pro-inflammatory chemokine, RANTES, which is a driver of chronic kidney disease. And so this pathway, which was reported in 2008, is discussing how paracalcitol can be used to attenuate renal inflammation, which is being driven by nuclear factor kappa B. More recently, it has been discovered that paracalcitol, also by stimulating vitamin D receptors, upregulates a key enzyme, an epoxygenase, CYP2J2, which puts epoxy groups on arachidonic acid, converting arachidonic acid, a pro-inflammatory lipid, into anti-inflammatory lipids known as EETs. In addition, stimulating vitamin D receptors with paracalcitol is now known to upregulate the master anti-inflammatory antioxidant transcription factor, NRF2. Upregulation of NRF2 in turn upregulates GPX4, glutathione peroxidase 4. This enzyme takes an electron and energy from glutathione to chemically reduce reactive oxygen species as well as peroxidated lipids, which are the upstream drivers of the programmed cell death pathway known as ferroptosis. So putting these pathways together, this is just a subset of the many benefits that vitamin D receptors generate when they are stimulated. It's important to discuss now the importance of suppressing renal inflammation in the context of autoimmune diseases. And this paper from 2022, the role of exposomes in the pathophysiology of autoimmune diseases, pathogens. What is an exposome? The exposome concept refers to the comprehensive measure of all environmental exposures, physical, chemical, biological, and social, an individual experiences over their lifetime, from conception onwards, and how these factors impact health. It acts as a counterpoint to the genome, aiming to explain the environmental causes of disease. This would include chronic viral infections, including the deposition of viral toxins in organs, sometimes locally produced, in other cases remotely produced, and then brought by physiological process such as circulation to the organ. Here we're talking about the kidneys. And this figure from the paper shows elevations in IgA antibody against 29 different human tissue antigens in the blood of patients with mild to severe SARS-CoV-2 infection in comparison to healthy controls. So these are many proteins that are structural, involved in tissue integrity, as well as hormones. So this means that the COVID-2 infection is producing immune responses that generate autoimmune conditions from a wide range of autoantigens. This is an example of an exposome. The COVID-2 infection creates an environment where the immune system becomes sensitized, and through molecular mimicry as well as other mechanisms, autoantigens are now recognized inappropriately as foreign agents. And from that point on, the establishment of autoantibodies, the person now suffers from these autoimmune states, which the upstream etiology was a pathogen infection, a microbial infection. So in the case of spike protein, the S1 fragment accumulating in the kidneys, a modified hypothesis, spike protein S1 fragment is likely to provoke chronic renal inflammation, and by doing so, sets the stage for the possible production of autoantibodies, leading to chronic autoimmune disease. Now we'll take these concepts and move towards a specific autoimmune disorder, IgA nephropathy. and we'll discuss this in the context of immune complexes. Pathogenic immune complexes are formed when antibody-antigen complexes accumulate, fail to be cleared, and deposit in tissues, inducing severe inflammation and damage. Immune complexes are key drivers of type 3 hypersensitivity, systemic lupus erythematosus, glomerular nephritis, and vasculitis, often resulting in neutrophil-dependent tissue damage, complement activation, and fibrinoid necrosis in small vessels, joints, and kidneys. Some key papers searching on PubMed for the concepts of immune complexes in COVID. There's 130 papers. One of these papers deals with platelet-virus interactions, the platelet-virus axis in human disease. This is a very important recent review which bears on the issue of long COVID microclots. Two additional papers, immunoglobulin G complexes from post-infectious ME/CFS, myalgic encephalomyelitis, chronic fatigue syndrome, including post-COVID ME/CFS. One additional paper from this search on PubMed, immune complexes in COVID, this one is a vaccine injury paper. Vaccine-induced immune thrombotic thrombocytopenia, an immunopathogenic model of dysregulated vaccine-triggered immunity. In the latter part of this YouTube, we are going to discuss immune complex-mediated renal disease, particularly IgA nephropathy. And there are IgA nephropathies that are new onset cases after COVID infections. And the issue of new onset IgA nephropathy after COVID vaccine also needs to be discussed. I would like to recommend a blog from the telemedicine group Rhythm. A link to that blog is highlighted in blue. Long COVID immune dysfunction, why your immune system stays activated. This blog connects to the review at the top right of the slide. Mechanistic insights into long COVID, viral persistence, immune dysregulation, and multi-organ dysfunction. Now, reading from the blog on Rhythm, one of the leading mechanisms driving long COVID immune dysfunction is the concept of viral persistence. Initially, medical science assumed that SARS-CoV-2 was completely cleared from the body within a few weeks of the acute infection. However, recent studies have proved that the virus, or fragments of its RNA and proteins, can hide in host tissues for months or even years. These hidden viral reservoirs have been identified in the gastrointestinal tract, the central nervous system, lymph nodes, and cardiovascular tissues, according to a 2024 study published in MDPI Viruses. These lingering viral components act as a constant source of antigen stimulation. Because these viral fragments are continuously shedding into the bloodstream, the immune system is never allowed to rest. It constantly detects the presence of a pathogen and mounts an ongoing inflammatory response to fight an infection that it cannot fully clear. This continuous stimulation drives the systemic myeloid inflammatory responses that disrupt cellular homeostasis. The gut is a particularly common reservoir. Biopsies have revealed persistent SARS-CoV-2 RNA in the mucosal tissues of many long COVID patients, directly correlating with the severity of their ongoing systemic symptoms. It is important to apply these concepts to the kidneys because it is now known that the CoV-2 spike protein, its S1 fragment, becomes highly localized in kidneys. The impact of the S1 fragment in the kidneys is likely connected with renal inflammation as well as tissue damage. We have been emphasizing that inflammation and damage to proximal tubules will result in a loss of calcitriol biosynthesis and secretion from the kidneys. Calcitriol is the most powerful immune hormone in the body. It plays critical roles in anti-inflammation and antioxidant capacity of many tissues. Besides damage and loss of proximal tubules in COVID-impacted kidneys, it is also important to focus on peritubular capillaries. These are small diameter capillaries that surround each nephron within the kidney. This paper is from 2020. Peritubular capillary rarefaction, an underappreciated regulator of chronic kidney disease progression. What is rarefaction? It means the lessening of density of tissues, especially of nervous or bone, but the same is true for the kidney. Peritubular capillaries resorb solutes that are transported across the proximal tubule epithelium as well as other parts of the nephron. In addition, secretory products that are coming from nephrons, such as calcitriol, are imported into peritubular capillaries where their circulation brings these hormones to the rest of the body. We are drawing on two previous YouTubes published on this channel, one in 2024, vitamin D receptor agonists for infectious disease vascular repair, and an older one from 2021, vitamin D receptor and toll-like receptor therapeutics for post-viremic COVID. Now going back to this paper from 2020, reading the abstract. Peritubular capillary rarefaction is commonly detected in chronic kidney disease, such as hypertensive nephrosclerosis and diabetic nephropathy. Moreover, peritubular capillary rarefaction prominently correlates with impaired kidney function and predicts the future development of end-stage renal disease in patients with chronic kidney disease, CKD. However, it is still underappreciated that peritubular capillary rarefaction is a pivotal regulator of CKD progression, primarily because the molecular mechanisms of peritubular capillary rarefaction have not been well elucidated. In addition to the established mechanisms, reduced pro-angiogenic factors and increased anti-angiogenic factors, recent studies discovered significant contribution to the following elements of peritubular capillary loss. 1. Prompt susceptibility of proximal tubule capillaries to injury. 2. Impaired proliferation of proximal tubule capillaries. 3. Apoptosis and senescence of peritubular capillaries. And 4. Pericyte detachment from peritubular capillaries. Sidebar comment. Pericytes are smooth muscle-like cells which surround small diameter blood vessels such as capillaries. Mainly based on the recent and novel findings in basic research and clinical study, this review describes the roles of the above-mentioned elements in peritubular capillary loss and focuses on the major factors regulating peritubular capillary angiogenesis, the assessment of peritubular capillary rarefaction and its surrogate markers, and an overview of the possible therapeutic agents to mitigate proximal tubule capillary rarefaction during CKD progression, chronic kidney disease progression. Proximal tubule capillary rarefaction is not only a prominent histological characteristic of chronic kidney disease, but also a central driving force of chronic kidney disease progression. In this regard, proximal tubule capillary rarefaction is a probable mechanism for long COVID. Here is a graphical abstract of concepts that are derived from papers we have been discussing in this YouTube, as well as concepts that we will discuss in the subsequent YouTube. Connecting A to B. COVID-2 infections result in the generation of multiple toxins, including the S1 fragment of the spike protein. We will refer to that as the S1 toxin. Why did the S1 fragment preferentially accumulate in the mouse's kidneys? There are probably a variety of mechanisms to account for this. I would like to now discuss a well-characterized pathway for transport of solutes to renal tissues, which is infrequently discussed. Let me briefly describe some of the tissue architecture that deals with this process. In panel B, we see two types of nephrons, a cortical nephron as well as a juxtamedullary nephron. The cortical nephron, its loop of Henle, is primarily located within the cortex of the kidney, the outer portion of the kidney. The loop of Henle for the juxtamedullary nephron is primarily located in the medulla part of the kidney. Both the cortical nephron and the juxtamedullary nephron empty into a collecting duct. Around each of the nephrons is a network of capillaries known as a peritubular capillary network. These peritubular capillaries play critical roles in resorbing solutes that are emitted from the nephron. And the nephron's key role is to resorb useful critical materials from the urinary filtrate and transport them into the interstitial space that lies outside the nephron. Then these solutes, as well as water, are resorbed by the peritubular capillaries and then return to the circulatory system as a whole. So how does this process work? Are there unique aspects of peritubular capillaries which facilitate this transport? The answer is yes. Endothelial cells of peritubular capillaries contain numerous pores known as fenestrae. Fenestrae is Latin for windows, and we will take a look at the ultrastructure of these fenestrae in the next slide. The fenestrae of peritubular capillaries are the major pathways by which solutes and water in the interstitial space surrounding nephrons is resorbed back into the circulatory system. What about the reverse pathway? Can a viral toxin such as the S1 fragment of the spike protein during a COVID-2 infection pass from the serum into the interstitial space via these fenestrae? That's one key question. An alternative way that the spike protein S1 fragment toxin could move from the serum into the interstitial space to impact the nephron directly would be by disrupted endothelial tight junctions. And this disruption of endothelial tight junctions could be formed in response to COVID toxins as well as oxidative stress. The reason to address these issues is to help explain the intense localization of the COVID-2 spike protein fragment, the S1 fragment, in the kidneys. Is this due to aberrant permeability of peritubular capillaries? Do they enter the interstitial space of the kidney via the side doors to nephrons, i.e. the fenestrae located in the peritubular capillary networks? This is an ultrastructure view of what the fenestrae look like. This is from a paper published in 1985. This is a scanning electron micrograph of a specially prepared preparation. One sees the circular fenestrae, which again is Latin for windows, with a number of filaments arranged in a hub and spoke fashion. The arrangement of these filaments is known as a diaphragm. Note that the diaphragm has numerous holes. The fenestrae have a diameter of about 60 to 80 nanometers, whereas the holes in the diaphragm are about 6 to 12 nanometers in dimension. It is through these pores that solutes and water pass from the interstitial fluid into the serum. Vice versa, solutes in the serum can pass through these pores, if they're small enough, from the serum into the interstitial space, where they can affect the physiology of the nephrons. And it's this back and forth exchange which provides the body with a key element of the endocrine system. The passage of calcifidiol and calcitriol, both are secreted by the proximal convoluted tubules in the kidney. Conversely, inflammatory mediators that are present in the serum, if they're small enough, can pass through these diaphragms, the holes in the diaphragms, enter the interstitial space surrounding nephrons, and thus alter the physiology of the nephron, potentially changing the biosynthesis of hormones such as calcitriol biosynthesis. Now, the movement of solutes, either from the interstitial space to the serum or in the reverse direction, is governed by what are called Starling forces, named after a physiologist, Starling. They are composed of hydrostatic force as well as an osmotic force, and they are expressed in this classic equation. So the diaphragms of fenestrated endothelia are gatekeepers of vascular permeability and blood composition. So while describing the dynamics of microvasculatures in various organs and how they're impacted by COVID and long COVID, it is necessary to go down to this level of analysis to ask questions about how a disease state is impacting the fenestrae and their diaphragms in specific organs. In this case, the microvasculature of the nephrons. In the upper right of the slide is an analogy which can be used to help describe the physics that take place in these fenestrae and their diaphragms. Consider this window in a cathedral. It is known as a rose window. Imagine taking the panes of glass out from the radiating spokes, but leaving the glass in the center of this hub and spoke arrangement in place. That would mimic analogy to the diaphragms in the fenestrae inside a renal peritubular capillary. With these pathways opened, gas flow could occur in either direction through this cathedral window, in and out of the cathedral's interior. That would be analogous to solutes and water moving in and out of the serum and the interstitial space, back and forth. Large molecular weight proteins that are present in the serum, such as albumin, which has a molecular weight of about 66 kilodaltons, is too large to pass through these pores. And so, realizing this, one can appreciate how evolution has designed a key structure which helps maintain the osmotic pressure of the serum, and it's known as the oncotic pressure, which is a colloidal pressure associated with large globular proteins. So in a normal, healthy system, albumin is blocked by this hub and spoke arrangement of filaments, the diaphragm, from passing from the serum into the interstitial space. However, in disease states, it might be possible that these diaphragms become injured. That's an open question. Certainly, the expression of key proteins are known to be necessary to form these diaphragms. But in the case of COVID and long COVID, we have to look at alternative pathways by which permeability of capillary beds, peritubular capillary beds around nephrons, could be potentially altered. And that is to answer the question, again, how does the toxic CoV-2 spike protein accumulate in kidneys? Can the toxic CoV-2 spike protein S1 fragment, which has a molecular weight of about 75 to 125 kilodaltons, depending on its glycosylations, can this S1 fragment pass through injured fenestrae in renal peritubular capillaries? That question can be addressed by looking at the molecular weight of human serum albumin, which is 66.5 kilodaltons. So human serum albumin is in high concentration in the serum, but in low concentration in the interstitial fluid surrounding nephrons. This is because of the diaphragms in these fenestrae. They block the large molecular weight globular protein, human serum albumin, from passing through the fenestrae. So if the fenestrae became injured and the diaphragms were lost, it would be possible that human serum albumin could pass directly from the serum to the interstitial fluid of the kidney. But at this point in time, there's no direct evidence of that. It's just a hypothetical condition at this point in time. Much more probable is that the spike protein S1 fragment during a COVID infection circulating in the serum could pass through disrupted tight junctions in peritubular capillaries. These tight junctions hold the endothelial cells together, and under oxidative stress or the toxic actions of viral proteins, spike protein S1 fragment, or the CoV-2 envelope protein, or other toxins, the loss of these tight junctions would open very large pores that would allow components of the serum to enter the interstitial space. Loss of tight junctions in the endothelial cells and/or loss of diaphragms inside fenestrae would produce serious permeability problems in peritubular capillaries, leading to disruption of renal functions. However, if the fenestrae and their diaphragms remain intact, these pores in the peritubular capillary surface are going to be playing critical roles in the pathogenesis of COVID and long COVID, as well as its treatment. Reading from the information vector at the bottom of the slide, here is a set of small molecular weight signaling molecules. Some of them are pro-inflammatory, others are anti-inflammatory. Others are key hormones or hormone analogs, synthetic versions of endogenous hormones. Calcifediol, the liver vitamin D metabolite, an active hormone that can activate vitamin D receptors. Calcitriol, the kidney metabolite of vitamin D, the most powerful immune hormone in the body. A synthetic version of calcitriol, paracalcitol, 125 vitamin D2, the preferred therapeutic agent to treat calcitriol deficits in renal patients. Arachidonic acid, formed by the actions of phospholipase A2, a pro-inflammatory lipid. It is upstream of the production of three classes of pro-inflammatory molecules: prostaglandins, leukotrienes, and thromboxanes. Arachidonic acid can be modified by epoxygenases to become EETs. EETs are anti-inflammatory. They also promote healing of injured tissues. So all of these molecules within the set, because they are small molecular weight compounds, they can easily pass through these pores, the fenestrae, and their diaphragms in renal peritubular capillaries. And because of that, this transport is going to be critical for pathogenesis as well as healing. And so the discussion of therapeutic agents is going to come back to these structures, and we will address that in future YouTubes. If you would like a PDF slide summary of this particular YouTube, I invite you to visit a Google Docs folder. The URL is at the bottom of the slide. If you navigate to a subfolder, Download First, you'll be able to locate the PDF slide summary of this talk more easily. These PDF slide summaries of the YouTubes on this channel contain active links so that you can navigate more easily between the YouTubes on this channel. You're welcome to download all of the material on the subfolders in this Google Docs folder.